we are. I think we have to hurry up a little bit. So maybe we can move to the third topic, which I'm going to talk about a very exciting news on Kymera's STAT6 PROTAC. So PROTAC has been a hot topic for our group. We talk about that for a while. Even beyond Kymera, there are some other companies has been focused on PROTAC. Um, you know, but why is STAT6? So STAT6, PROTAC has been mainly focused on cancer, but the data so far is not that great. So that's why there's another direction to target, molecules around inflammatory disease. So STAT6 is one of the transcription factor driven allergic type 2 inflammatory response. So the upstream is Th2 cytokine, for example, IL-4 or IL-13. So for example, as I show here, IL-4, IL-13 actually share the same subunits, IL-4 receptor alpha. There is a common gamma chain for IL-4, and IL-13 have another receptor. Both cytokines share IL-4 receptor RA. So the Dupixent, another name is Dupilumab, which target IL-4RA, which can block both cytokines, IL-4 and IL-13. So that has been very successful drug, for the field. So why there is a unmet medical need for STAT6? STAT6 is a very specific transcription factor drive the downstream, and then this pathway has been clinically validated for Th2 allergic responses and atopic diseases. And STAT6 actually there's some genetic association, which has been demonstrated. And then again, why STAT6 inhibitor, small molecule inhibitor is interesting because antibody is great. You might have a very long half-life, but it's very expensive. So if you have oral delivery potential, versus you have to have patient has to go to the hospital to give the injection. So of course that would be more convenient and economically more friendly. So this is next slide. Another slide. So basically, the Kymera, this company has basically have done the investigation and also the market, investigation. So if you really talk about Th2 related disease, AD and many others, even asthma, COPD, so the patient will be like 140 million patient, but however, so far only 2 million patient has been injected. So that means you have quite a lot of patients which did not give the therapy. For example, Dupixent has not been covered. You have a very huge population. People has not been given any of the treatment. So if you have something very convenient, very cheap, like oral inhibitor, for example, it might have the potential to transform the treatment paradigm for this disease. That is something quite interesting. Another slide. So that's why they have been thinking about PROTAC for STAT6. So the idea is have a molecule binds to STAT6 and also E3 ligase to drive degradation. So again, this PROTAC has to be, this is a very big molecules. That's why for cancer setting, it's not very efficient actually. But whether there's any benefit for STAT6, that is something people didn't know, especially targeting transcription factors. Can you move forward? Thank you. So they have done the PROTAC for STAT6, and then they have done specific analysis, the specificity analysis. What they found is this STAT6 inhibitor actually is specific targeting STAT6, not other STAT1, 2, 3. So this is really STAT6 inhibitor. And also they have done different cells, immune cells, and also beyond immune cells. They have done quite a lot to show this molecule really inhibit T cell or B cell monocyte responses related to the cytokines. And also they have done the comparison with with antibodies. So the EC50 is even better. So in animal models, preclinically, they also show quite impressive efficacy in Th2 related, HDM is what kind of Th2 some kind of allergic response models. One slide. So clinically, what's really interesting and also I think is very impressive is for most small molecule inhibitors, whether you can target the response in the blood or really in the tissue, that can be the problem. Especially, for example, antibody, right? It has a very easy penetration to get delivered in the circulation. But however, to the tissue, whether it has a response, always have a weaker response. So, but in this case, the small molecule inhibitors, so STAT6 degrader, sorry, and then shows very similar efficacy to degrade STAT6 in the blood versus skin. And also they have seen some very quick, like in 14 days, as data show here, in 14 days, you have a maximum reduction of the Th2 cytokine. For example, track is another name is CXL17 is kind of Th2 chemokine. So in 14 days, in 2 weeks, you see great reduction up to 40% or 60% of different cytokines. One slide. So, and another thing is how about comparison with the Dupixent in the clinic? So of course the time is important. The early data they showed the reduction actually is even a little bit better than Dupixent. But the timing, this is a timing not consistent, but the early data showing is quite promising and even better. One slide. So another thing is they did see clinical benefit as the data show here, the reduction of the symptoms and also the clinical yeah, changes versus baseline. And also they have done the IHC to measure STAT6 activities by 29 days. The STAT6 IHC has been greatly reduced. So as I mentioned before, so reduction in the tissue is very hard by antibodies um, driven approaches. So then these small molecules degrader really see some tissue related reduction. That's quite interesting. And also, STAT6 and also some kind of cytokines, even IgE, that's something quite impressive. One slide. So again, by day 29, they also compare similar efficacy of these molecules compared to Dupixent. This is the same day 28 or day 29. And these are large cohorts from phase 3 trial of Dupixent. So most of the data actually is better than Dupixent. This is a 1 month, almost 1 month at week 4. So, and also very good safety signal. One slide. So based on this data, I think the stock has been increased quite a lot. This is the phase 1b trials, and they are going to start the phase 2 trial. And then for those with different dose ranging, and also they also have started the asthma trials, and hopefully, they can get much better data. I think in maybe next year, actually this year, someday they also have asthma data coming out. And the next year they are going to have AD phase 2b data for 2027. I hope those programs will get successful. One slide forward. So why STAT6 degrader data looks so strong? Actually, beyond STAT6, I'm going to introduce other programs. So actually, STAT6 is not the first program for Kymera. They have other programs. Why this program is getting so interesting? I think there is something unique for STAT6. First of all, this is a transcription factor. In most cases, transcription factor has to enter the nucleus, and the small molecule getting to the nucleus might be hard, right? But this molecule actually always trafficking, shuttling between cytoplasma and the nucleus. That is something unique for STAT6. And also, small molecules always have better exposure in the cytoplasma. So that's why if a molecule trafficking a lot, that might have a better target because you have a repeated access to its ligase. That's one thing I think might be not unique, but might be interesting for STAT6. And of course, STAT6 is very specific. And they also have a very druggable pocket, and also have they can really dwell onto put on like park onto the DNA. The dwelling time is not very strong, so that's why they always trafficking around. So it's easier to pull off. And also the protein half-life is short, it's better to be degraded. And also you have better degradation activities. And again, biology is very clean, very impactful. Maybe those are some of the benefit for targeting STAT6. If STAT6 is successful, then we have much more molecules to be targeted in the cytoplasma or in the nucleus. So that might open a new chapter. So this is why something very interesting, because it's one of the very first uh, PROTAC for autoimmune disease. And again, there are many more transcription factors which we did not target before, but it might open a new chapter. So one slide forward. So beyond STAT6, so there are other programs, for example, the IR5, and again, potentially they might target many more molecules. There's an open field of studies. And the IRAK4 actually was one of the very early programs for Kymera, and several other companies working on already partnered with big pharma. So right now, STAT6 is their leading program. One slide forward. So I think IRAK4 is something really targeting toll-like receptors or IL-1 pathways, important for inflammation, but it's more broad. Th1, Th2 is a master regulator. So then it could be very potent, but the problem here is whether it's a good target, better target compared to STAT6, whether, for example, infection, some of the side effects is not clear. But so far, I think data from STAT6 is quite interesting. And also there are beyond STAT6, there are some other molecules like TYK2, which later time we're going to talk about. So in general, I think this is a very exciting field of studies, and then might open a new chapter to target transcription factors or cytoplasmic membrane the proteins, which of course biology is very important. And there we have to really talk about the target biology and also how we can target this protein in a more effective way, whether PROTAC is the right molecules or other molecules. So I think that's very exciting to the field. So I can stop here if you have any questions.
Actually, there's a
Any?
question in the Q&A. Which E3 ligase for KT-621? Do you know that?
No, I don't know. I don't know which ligase. I haven't looked into that details so far.
Or Sean's question, what's your assessment on this potential of pipeline in a protein? Can this STAT6 become like a Dupilumab, become a pipeline
Yeah,
for multiple disease?
that's a very good point. So as I showed in the beginning, right? So there are the TH2-related. Most people just talk about AD, but I think for other disease like COPD or the others, could be quite interesting. This is just one of the AD is more like low-hanging fruit for TH2. And for asthma, right? Asthma, I think this Kymera already actively working on. So let's see. Let's wait and see. It could be beyond which the most antibody has been tested. So that's what I think is very interesting. And also one more thing is for this program, safety is very interesting.